Finally, a bit of a sunny day that almost resembles summer. I'm standing here in the ROPOS tool basket, which will go down on the next dive to recover some equipment that we've prepared on the bottom. A time-lapse camera system called Tempo Mini, which has been built by our French colleagues at Ifremer, and the bar's high-temperature thermistor, which has been recording temperature in the black smokers. So both of these instruments have been sitting down there since last fall, and during the last dive we removed them from where they were deployed, brought them over to an area and have them all staged and ready for recovery. So on this next dive we're going to lift ROPOS up in the air and lower it down onto the top of the tool basket here, latch into this point, and then the sub will take the tool basket in its belly hook all the way down to the seafloor, put it down on the seafloor, and then run off and get the Tempo Mini camera system, pop it in the basket, pick up the bar's temperature recorder, pop that in the basket, and then use the manipulators to essentially pull these straps over and secure everything for recovery. Then at that point, ROPOS will fly up and then descend down on top of the tool basket and once again latch into this hook and bring the whole thing back up to the surface, where we have a lot of excited people waiting for instruments and any animals that have grown on the instruments. We've discovered that the instruments we put down there are great surfaces for animals to colonize. And not only that, but we have a date, so we know when these things came. So we've got a couple people in the lab already working on juvenile tube worms that were recovered from the water sampling system that we recovered on this last dive. So here in the lab, we've got Françoise and Lara who are looking at an unexpected sample that came up on the RAS sampler itself. We've got a bunch of, what have we got in this container here?
It's limpets.
Limpets, tiny little snails that are a couple of millimeters in diameter. They're actually hundreds of thousands of these things per square meter around most of the hydrothermal vents. And in here, we've got some large buck-sinned snails. So these are deep sea snails that live normally outside of the hydrothermal vents, but they're also fairly tolerant of mild high temperature fluids. So they're actually hydrothermal fluids. So they actually come into the vents and graze around the periphery of the vents on the bacterial mats.
And tube worms in the cold room.
And in the cold room. So let's go into the walk-in cold room here, where we have a large collection of juvenile tube worms that came up on the sampling cap from the RAS. It's quite cold in here. We're down at 4 degrees Celsius. So in here we have a dissecting tray full of juvenile tube worms. These had colonized the sampling cap that goes over the vent since last September. So you can see these are all individual worms in these tubes, and in some cases you can see the red you can see is the tissue part of the worm that's sticking out of his tube. These are their gills. So these are all babies that are less than a year old, and we're quite interested in knowing where the larvae that grew up to be these tube worms came from. So Lara, who we were talking to in the other room, is going to be doing a study of the population genetics of these tube worms. We're trying to understand, this is a marine protected area at this site, and we're trying to understand where are the important nurseries and sources of baby tube worms within the area of the marine protected area. What kind of habitat do they favor? So we're looking at the genes of all the babies we can find to determine whether they come from a single source or from many different sources. If they come from a single source, then we have to be very careful about protecting this, even from scientific sampling. It's getting too cold in here, let's get out.